Landfills are the third most important methane source, anthropogenic methane source. And in landfills, methane is produced from the bacteria inside of the landfill. But luckily, there are like other groups of bacteria that can oxidize, remove, eat up all these methanes, living outside of the cover soil where oxygen is present.
Something to Chew On is a podcast devoted to the exploration and discussion of global food systems. It's produced by the Office of Research Development at Kansas State University. I'm Maureen Olevnik, Coordinator of Global Food Systems.
I'd like to welcome back our co-host, Jim Stack, Professor of Plant Pathology in the College of Agriculture, and welcome our newest co-host, Dr. David Proctor, Emeritus Professor of Communication Studies in the College of Arts and Sciences here at K-State. Destructive greenhouse gases are generated in many areas of the food system, from hay bales to landfills, plastics to water waste. An intersection of engineering and microbiological sciences has allowed Dr. Jeongdae Im to tackle multiple challenges in studying ways to mitigate the negative effects of those greenhouse gases. Welcome, Dr. Im. Can you start by telling us a little about yourself and how you found your way here to Kansas State University?
I originally from South Korea, and I actually got my bachelor's and master's from Seoul National University in Seoul, South Korea. And I spent 2 years, mainly because my GPA from my undergraduate was not good enough to get into a good schools in US. So I spent 2 years in the research institute in one of the major construction companies in South Korea. And then I got into graduate program at the University of Michigan, and it took me 4.5 years to get my PhD. And I met my wife at the University of Michigan, who got her PhD in psychology. And I got a postdoc position at the University of Tennessee, and I had an additional position at the Oak Ridge National Laboratory. And I was a civil engineer by training. I got all my degrees, bachelor's, master's, and PhD degrees from civil engineering, but my postdoc position was in microbiology. And I became more serious in microbiology, although I used microbiological technique for my degrees. So I stayed in Tennessee for 4.5 years. And by the time that I moved out from Tennessee, my wife got a tenure track position at the University of Massachusetts, where I believe that Dr. Stack got degrees.
Yeah, 2 degrees here.
Yeah, 2 degrees from there. Although I had tenure track position offer from New Mexico State University, when my wife got an offer from the University of Massachusetts, we were a little too naive. I gave up my offer from the New Mexico State and accepted research assistant professor position at the University of Massachusetts. And after 2 years, we realized that the University of Massachusetts will not gonna give me a position until I get a decent amount of grants or something. So we went out to get additional positions, and I got a position here at Kansas State, and my wife got a position at the University of Texas in Dallas. And since the two universities are in the same time zone, which was important for us. And then in addition to that, we had a direct flight from Manhattan to Dallas, which was one of the major reasons that we decided to settle down into a new universities. My master's and PhD topic was about greenhouse gas emissions from landfills. So I studied microorganisms that producing and reducing greenhouse gases, including methane and nitrous oxide. For my postdoc, the major research project that I took over was to examine the environmental fate of emerging contaminants, including bisphenol A and fluorinated compound. And then another research topic that I learned when I was at University of Massachusetts, well, I went to one of those seminars hosted by the biochemistry departments. And 2015, the Department of Biochemistry at the University of Massachusetts inherited plant cell culture library from Monsanto. And they were really excited about these donations. And I went to one of those seminars and learned that I may be able to utilize those plant cell cultures in my research to investigate the plant bacteria interactions. So that's one of my current research thrusts that I'm currently working on. So since my wife is in Dallas, I drive from Manhattan to Dallas like twice in a semester. Once the winter or summer vacation starts, I go there. When we have a longer vacation, like spring break or Thanksgiving, I drive there. So when I drove from 7 hours drive from here to there, the most unfamiliar sceneries that I saw was a lot of hay bales. Although at that time, I didn't even know the name of these hay bales. And since my research was on the greenhouse gas emissions from organic matter, organic waste in landfill, I thought that the hay bale is a big chunk of organic that may produce a lot of greenhouse gases. So I went back from the trip to Dallas and looked up some literatures to see if there's any previous research on greenhouse gas emissions from forage conservation, and then found out that there's no research done on that topic. And I was really excited because I knew that it's going to fly. So I did some preliminary experiments with the alfalfa sample that I got from my colleagues at the Department of Agronomy, and basically incubated the alfalfa sample in the absence of oxygen, simulating the silage process, and monitored the gas productions and compositions. And I found out that significant amount of nitrous oxide can be produced from this silage process. So I submitted proposals not only to NSF, but to GFS. And then I got the app score, the seed grants from NSF, and also got the GFS seed grants, which were really, really, really helpful to move forward. And then I did more experiments with different crops, including alfalfa, corn, silage, which are the three major crops used for silage, and then replicated the results. A lot of nitrous oxide was produced. And I submitted another proposal to NSF, the career program, and was notified that I got the awards. So those are things that I'm currently doing, and I'm still interested in examining the environmental fates of emerging contaminants and how to facilitate microbial processes to remove these contaminants, including plastics and other contaminants.
Well, that's a pretty big challenge. No, there's a lot to do in that arena, an awful lot to do. And I think it sounds like you've given this a fair bit of thought. Some of the challenges that we have are that they're linked. And so trying to solve one problem oftentimes exacerbates another. I was wondering if you encounter that dilemma as you're trying to clean up plastics. I just wondering what your thinking is on that.
Yeah, so that dilemma was actually one of the starting points of one of my research projects. It is not related to the plastics, but landfills are the third most important methane source, anthropogenic methane source. And in landfills, methane is produced from the bacteria inside of the landfill. But luckily, there are like other groups of bacteria that can oxidize, remove, eat up all these methanes, living outside of the cover soil where oxygen is present. And in order to facilitate their activities, we typically provide nitrogen source. They need major elements like carbon, nitrogen, phosphorus, right? So just like plants. So nitrogen amendment is one of the most widely accepted strategies to facilitate methane oxidizing bacteria. But the problem of that specific strategy was that nitrogen amendments not only stimulate methane oxidizing bacteria, but also stimulate other groups of bacteria called ammonium oxidizers. Not sure that you know, but when farmers apply nitrogen to the soil, 50%, the half of the fertilizers are lost, mainly because of these activities of this ammonium oxidizing bacteria. And they oxidize ammonia to nitrate, and nitrate can be leached out to the groundwater, or it can be further converted to nitrous oxide, which is the third most important greenhouse gas, which is why the soil management fertilizer application is the major source of nitrous oxide emissions in general. So in landfill projects, our hypothesis was that if we add nitrogen together with other chemicals that inhibits ammonium oxidizer, we may be able to remove methane and simultaneously repressing nitrous oxide. So the project was funded by DOE, and we had a dilemma. When we apply nitrogen, we can minimize methane emissions, but there was a side effect, nitrous oxide. So that was the starting point of one of my projects. So at the end of the project, we were successfully demonstrate this combination can minimize greenhouse gas emissions from landfills, and we demonstrated it actually works in the real landfill. But the problem was that the chemical that we used was expensive, and people feel reluctant to apply, another chemicals in landfill. So yeah, there were like practical barriers to implement this technology in real world. And then I saw this paper published in 2013 or 14 in PNAS that some plants. So when we apply fertilizers, there is a competition between plants and this ammonium oxidizing bacteria. Ammonium oxidizing bacteria oxidize this nitrogen fertilizer to survive, and plants need this nitrogen. So in the paper that published in 2013-14 in PNAS, they found out that certain plants can produce certain chemicals, phytochemicals, that can inhibit ammonium oxidizing bacteria. It is called the biological nitrification inhibitors. So the researchers in agricultural area study this BNI, biological nitrification inhibition process to minimize nitrogen usage for fertilizer usage and maximize crop productivity by genetically modifying this function to produce more of these chemicals that can minimize ammonium oxidizer activity. I figured that I may be able to use the same plant instead of the chemical that I used in landfill. So basically, I got interested in studying this biological nitrification inhibitor to minimize greenhouse gas emissions. And I actually got the seeds that was used in the study that was published in PNAS and germinated and hydroponically grow in the lab as a side project, and did some preliminary study and was able to replicate the study. But the problem was growing these plants, intact plants, and use for study was really, really, really difficult. And I learned that researchers also struggle to avoid like fungal contamination, and it takes up a lot of space, and we need greenhouse to do this type of experiments. And then almost at the same time, I got into this seminar and learned that people use plant cell cultures, and plant cell cultures can potentially be used for this type of analysis for research instead of intact plant. So I'm trying to develop a high throughput screening method using plant stem cells and plant cell cultures and this ammonium oxidizing bacteria to screen this BNI activity and further examine this phenomenon.
So Dr. Im, you've got some phenomenal research going on in the laboratory. Do you have avenues in place at this point for doing that in a more applied manner, getting that information out into usable hands, or are we still at the very early stages of research in these areas?
So for the greenhouse gas emissions from forage, the project that was supported by GFS, at the same time, I was able to I tested different chemicals to see if we can minimize greenhouse gas emissions from forage, and found that certain chemicals, not expensive chemical, can reduce greenhouse gas emissions over 95%. And I actually filed a provisional patent last December, and I contacted this company who's the company Corteva, a Pioneer Corteva, which is the largest silage inoculant producing company in US. And they agreed to collaborate with me to co-develop these novel silage inoculants that can not only stimulate fermentation in silage, but also can minimize greenhouse gas emissions from. So they accepted my idea and decided to work together. It is moving forward. And the plant cell culture problem, the project is very early stage, and it is just proof of concept stage that can directly apply to the industry.
Wondering if you have been able to use any of the plants that you're growing to address the landfill issue, sort of reduce the greenhouse emissions coming out of that.
So the goal of this plant bacteria interaction project, the ideal picture to screen indigenous plants in different states. If we can come up with a screening method, we can select indigenous plants in different climate region and propose to each state that use this plant in your landfill that can minimize greenhouse gas emissions in your region. And in order to do that, without this high screening process, it'll be really tedious and takes a lot of time and effort to screen this BNI activity from different plants. So my goal is to come up with a screening method that can be used to select indigenous plant species that can be used in landfills.
Yeah, to follow up on that, so that strategy has worked in land reclamation, where they've been able to use plants to reclaim lands that have been used for strip mining and things like that. Landfills have a life cycle from when they first prepare the ground to receive waste, then they're adding waste over a period of years, then they cover it. And I'm trying to envision the strategy for using plants. The emissions begin almost soon after the waste is accumulating. So how would you use the plants during lifespan of the waste facility, of the landfill?
I'm really a little surprised that you know the life cycle of the landfill. So yeah, once landfill is finished, we cover not only with the soil, but with the membrane. We close the landfill with a membrane and add a lot of soil. I don't exactly remember the depth of the soil requirement. We cover the landfill with the soil, which is called the landfill cover soil. But the problem is that even with the membrane, things happen. The landfill gas leaks out of the membrane and emitted into the atmosphere. So we need to have a plan. And as Dr. Steck just mentioned, there is a life cycle of the landfill, and a significant amount of landfill gases are produced at the very first 5 to 10 years of the period. And then a smaller amount will be produced over 20 to 30 more years after the initial period. And currently, there's no regulation on how to vegetate this landfill cover soil. But when we go to the landfill, a lot of indigenous plants are growing on top of the landfill. So what if we can select and propose specific indigenous plants and just have it grow on top of the landfill, which can potentially minimize greenhouse gas emissions? That'll be just great. And at the very beginning of the landfill, we can provide nitrogen source to stimulate methane oxidizers only at the beginning, and let it go after the 5 to 10 years, active years. Then that'll be the big picture that I have in mind.
That sounds reasonable. Yeah.
And then these nitrogen fertilizers can be obtained from leachate treatment. The major concerns from landfilling is not only the greenhouse gases, but also leachate. Leachate is the liquid leaked out from the organic waste. And then it is typically sent to the local wastewater treatment plant. And nowadays, one of my colleagues in our department is currently working on the project that we can extract nitrogen fertilizer from the wastewater process, Dr. Parmesan, who was featured in previous podcasts, the Something to Chew On podcast. So we can extract nitrogen from the leachate and apply on top of the landfill to stimulate methane oxidizers. And these nitrogen amendments may lead nitrous oxide production, but we can repress the nitrous oxide production by indigenous plants that we can apply at really cheap cost. All we have to do is just get the seeds, and as far as we can screen which plant we can use.
Have you developed any relationships with municipalities or counties where landfills exist?
Yeah, so we are actually running reactors, 1 meter long, 30 centimeter in diameter cylindrical reactors to simulate the landfill cover soil. So basically we fill up the soil up to 1/3 of the reactor and then plant certain plant species and flow in the methane from the bottom and see how the plant can minimize greenhouse gas emissions from the landfill. And in order to establish these reactors, researchers typically use the soil from the landfill. So I contacted this Salina Municipal Landfill, and we are getting the soils from there. And we discussed the possibility to potential collaboration to demonstrate this technology in their landfill. So once we come up with the specific solution, the type of the plants that we can use and the nitrogen dosage and how to monitor it and everything, we can try to see if it actually works in the real landfill, in Salina landfill. Yeah, I have a collaborator in municipal landfills.
I was interested in material that was provided about your research. I was interested in the piece that talked about the patents that you received that you have applied to wastewater treatment facilities in Korea. And I was wondering if you could talk a little bit about that process and sort of how your research has impacted those wastewater treatment facilities.
So the technology that I patented on was the membrane bioreactor system that doesn't require additional setup to circulate the wastewater stream. So typically, wastewater treatment process relies on different tanks that's maintained under different conditions. So the most important process in wastewater treatment is to provide oxygen to convert the carbonaceous compound into carbon dioxide and biomass, and convert ammonia into nitrate. And then we send the water into another tank where this oxidized nitrogen is converted to dinitrogen, which is called denitrification. And then we typically have another tank where phosphate is accumulated into the bacteria so that we can remove the phosphate by removing the bacteria. So this process takes a lot of space and energy to move water from one to the other. But the technology that we developed was to use for local community where the government do not have much money or space to have large treatment systems. So it was compacted system that do not need the tanks where different microbial processes are occurring. So actually, I didn't do much follow-up work to follow up that study because my PhD topic was not about the wastewater, but about the greenhouse gas emissions. And my postdoc work also not about the wastewater. So I still know how things work in wastewater membrane technology used in the wastewater treatment process, but I haven't done much work to follow up.
You had mentioned, I believe, in kind of your opening comments, plastics, and that is a rather large issue right now, everything from microplastics up to containers. And I was just wondering what your thinking is on that, or what research you're pursuing for addressing that challenge. That's big.
Yeah, the plastic issue is really big. So the project that I worked on was the bisphenol A, also known as BPA. So when you buy a plastic product, many times you see this BPA free. So BPA is monomer used for basically a building block to make polycarbonate. So polycarbonate plastic is essentially the plastics, rigid and transparent plastics that is used for the glasswares, or nowadays we see a lot of this rigid, transparent plastic that are used instead of glasses. So BPA is one of the most produced chemicals in the world as a single chemical. So BPA is not only used as monomer for polycarbonate plastic, but also used as an additive in different types of plastics. Like, I don't remember the types of the plastics, but it is literally the most produced chemicals in the world, one of the most produced chemicals in the world. So the project that I worked on, funded by the BPA producers, was that since BPA is widely used, environmental fate of the BPA has been widely investigated for last couple of decades. So we now know that BPA can be degraded by bacteria, fungi, plants, and by sunlight. And there are many organisms and abiotic processes that are known to degrade BPA. But most of these degradation processes needs oxygen, and it only occurs on the surface of the water, like BPA degradation by sunlight. Over the years, people try to come up with a solution or degradation mechanisms in the absence of oxygen, but failed. There was no degradation process that can remove BPA in the absence of oxygen. It doesn't make sense because BPA is released into the environments, and not only released into the aerobic environments, but also released into the anoxic environments. And if there's no degradation process in the absence of oxygen, there is supposed to be a lot of BPA deposited in the anoxic sediment, but that's not the case, suggesting that there should be something. So the DuPont wanted us to investigate the BPA degradation processes in the absence of oxygen. So I tested different redox conditions, different sediment samples, different microorganisms that can remove BPA, degrade BPA in the absence of oxygen, and actually found out that certain groups of bacteria that can oxidize manganese produce this manganese oxide. And manganese oxide is typically settled down into the anoxic environments and deposited in the anoxic sediments. And it is known to be very reactive, and I demonstrated that this manganese oxide can remove BPA at very high rates. So this coupled biotic-abiotic mechanism may explain one of the degradation of BPA in the absence of oxygen in anoxic sediments. That's the project that I worked on. And this technology can be used in many other applications where BPA can be a problem.
That manganese, is that microbially mediated, or is that strictly a chemical reaction in the sediment?
So manganese oxide formation is mediated by microbial process, but degradation process, I think it is abiotic.
So that leads me to a question. So you've talked about plants, microbes, and engineering. Where do you get the students?
I get the students from civil engineering and teach microbiology and teach plants. And it'll be ideal to recruit the students from civil engineering who knows microbiology. That'll be ideal. But I need a student who has an engineering mind to find a solution and who is willing to study this other stuff. Yeah, it is really interdisciplinary. And I typically get help from my collaborators in plants and microbiology fields.
As you said, Jim, it's wide-ranging in the things that you're looking at. It's kind of shocking is the areas that you're looking at, going everywhere from microbes to microbiome type approaches all the way through the plant systems and into the engineering side of things. And one thing I mentioned as we started here that I found very interesting as well, that we've been doing presentations on the GFS seed grants over the last couple of days that we're going to be posting on our website. The focus on nitrogen contamination and greenhouse gas contamination as it's applied to plant materials was pretty strong in this last round.
where we had a faculty member from chemistry that was working with an agronomist looking at co-crystal formations with urea and applying that as a nitrogen, very specific nitrogen application to plants. We had faculty member out of chemical engineering that is working with a member in biology looking at the applications of nitrogen in corn root systems. And it just seemed, as you started talking about the work you were doing and doing the presentation that you did as well on the work with silage, there was something similar between those three where I could just see an intersection and a nexus between them. Am I thinking too broadly, or do you see something where there's a synergy there as well?
Yeah, there sure can be a synergy between this nitrogen cycle, nitrogen utilization, or nitrogen related microbial processes. One of the research that I'm currently initiate together with the faculty in this chemical engineering, Dr. Park Won-min, is to develop a genetically modified microorganisms that has embedded metabolite sensing system. So in order to investigate the plant-bacteria interaction, we have to set up additional experiments to monitor microbial activities or the effect on the plants. But if we can monitor the metabolites of the bacteria in situ or simultaneously, it'll be really beneficial to any research associated with the microbial process. The specific bacteria and the metabolites that we have in mind is ammonium oxidizer. And ammonium oxidizing bacteria convert ammonia to hydroxylamine and hydroxylamine to nitrite. And we are trying to make this genetically modified ammonium oxidizing bacteria that has a sensing system of two different fluorescence-based sensing system that can detect hydroxylamine and nitrite. So once we get that bacteria, we can do the similar gene modification on nitrifiers or the other nitrogen-related bacteria as well as a model to examine whatever microbial processes they are interested in. Basically, the BNI is the microplant interaction. Biological nitrification inhibition is one of the microplant interaction occurring in the rhizosphere. And I believe that Ryan Hansen is currently working on the project on the microbial processes in the rhizosphere. And the same concept can be applied to any microbial processes occurring in the rhizosphere, I believe.
I'm really interested in things that you said earlier about how nitrogen is used among farmers, but at the same time you have leaching that occurs, and that gets into the groundwater, gets into the streams, gets into the ponds. I'm wondering, are there things that farmers can do that can reduce the amount of that kind of contamination in their fields as they put down fertilizers? Are there processes already in place that can reduce that? Or is the research moving in that direction and we're just not there yet?
In the agricultural literature, there are many chemicals developed to repress these ammonium oxidizing bacteria. And the chemical that I used for my landfill study to minimize the greenhouse gas emissions was actually developed by one of those researchers in agriculture that was initially developed to minimize the activity of ammonium oxidizing bacteria. So yes, there have been many efforts to minimize the activity of ammonium oxidizers, to increase the nitrogen utilization efficiency and minimize the use of fertilizers. But unfortunately, the nitrogen utilization efficiency is still 50 to 60%. But I believe that this BNI, certain plants that can produce these chemicals that depress ammonium oxidizing bacteria, can be one of the solutions that can minimize the fertilizer use and maximize nitrogen utilization efficiency. It is very, very fascinating, the phenomenon that we should work on.
I think part of the problem in agriculture is the timing of the application. You have crops growing over a period of months, but the practicality of supply as it's needed. It can't be worked out for most crops. And so you end up having to apply it in batch. And so it's not utilized immediately. I think some of the solutions might be in some systems would be going to subsurface drip systems where you can supply the nutrient over time as the crop needs it. But again, that's a very costly technology to implement. And then the maintenance is nothing minor as well. So it's really just a challenge of how we do agriculture. In time, the value of food will likely go up, and making it more possible in some sectors to implement some of these technologies. It's amazing, though, that the reliance on engineering is only going to increase over the next 30 to 40 years for solutions to some of these big problems.
And the projects that I mentioned just a little bit earlier that had been recently discussed and recorded, those researchers came from all over the place on campus. They came out of, as I said, chemical engineering, civil engineering, agriculture, and it's most definitely going to work from all of those sectors to solve some of these problems to make it happen.
So, Jeongdae, where do your students go? So when they finish with your program, where do they work after that? Where do they go?
I only have 2 graduates so far, and one of them is pursuing PhD in other universities, and the other students got a job at the consulting firm. And the other graduating students is trying to get a job in consulting firm. And based on my previous experience with my colleagues, we get a job in local governments as an engineer to monitor the environmental health, or some of them got a job in national laboratory research institutes like Battelle. That's actually not a federal institute, or got a job in academia.
The graduate that got a job in a consulting firm, what sector of business was the consulting in?
Environmental remediation, clean up the contaminants in groundwater. That's a big market in our field.
Are there any other big challenges you want to go after? It doesn't sound like you're shy about taking on big challenges.
He's got a lot already that he's been tackling.
So the plastic contaminants in the ocean, until now, people typically concerns about the mechanical impacts on aquatic animals. Some birds tangled up in the strings, or the fish choked up with this plastic matter, but not much of a research has been done on the chemical impacts. So the plastics is made of chemicals, and they broke down by the sunlight and by the waves and by microbial enzymatic activities. But their chemical impacts has not been properly done. So for example, BPA is known to inhibit, based on my preliminary study, Rhodobacter is one of the most important microbial members in the ocean microbiome. And I have an evidence showing that BPA can inhibit this Rhodobacter. And then what does Rhodobacter do? Rhodobacter is responsible for carbon monoxide and sulfur cycle in the ocean. So it is possible that BPA leaked from this plastic debris may impact certain microbial groups in the ocean, may impact the global carbon and sulfur cycle. That's one of the projects one of my master students is currently working on.
That's a great one. And, the concern for the chemistry of the oceans is growing because of the impacts on coral systems and populations of various sea life. So yeah, that's a great one.
I wonder, in terms of your landfill research, as I've been thinking about this, thinking about sort of my own work when I was working with communities around the state of Kansas, in a way, it seems like you have sort of 2 potential tracks. They're not inconsistent, but one only has so much time. Is your focus on the landfill work, is it more national? So you're looking for these indigenous plants that can help mitigate greenhouse gases. Is it national, or would it make more sense to focus initially on what's happening in Kansas so that the scale would be smaller, but there could be faster impact because the number of indigenous plants that you might be looking at there'd be a smaller variety of those to check out rather than looking at that nationally.
My goal on this landfill research is to develop a high throughput screening method to detect the BNI producing plants. So if we can do that, such a technology can be used in everywhere in the world, in different regions. Let's say that in South Korea has a lot of landfill as well. And the plants that present in South Korea is totally different from the plants in US, in everywhere, right? So we can apply those techniques in South Korea and propose these indigenous plants that can be used in landfill, right? So once we have this technology, we can screen the plants that can be used in landfill anywhere. And also this technology can be used in agriculture research as well, because people are trying to develop genetically modified plants that can produce more BNI. And now the only method that can test this ability is to use the intact plant. But if we can do this with the plant themselves and high throughput manner, the whole process can be done in really easy and quickly and conveniently. So yeah, I'm trying to develop a method, the high throughput method, using not intact plant, but the plant stem cells. So such a technology can be used in many applications, and landfill is just one of the examples.
I can think of another area of need, and that would be feedlots and the waste that comes out of feedlots. Jeongdae, I enjoyed the discussion immensely and wish you well with your program. I think it's great that you're exploring different ways to solve some of these problems. So thank you.
Very ambitious research that is tremendously important. Very interesting.
The first two sentences that I used for my career proposal, so it was, what gets measured gets managed. And the first step to mitigate greenhouse gas emissions is to know where the sources are. And that the same concept can be applied to any environmental problems that we have or will have in the future. So my goal is to not only come up with the solution, but also to identify the problem.
Well, thank you so much, all of you. And we look forward to getting this posted and out for others to hear.
If you have any questions or comments you would like to share, check out our website at ksu.edu/research/global food and drop us an email. Our music was adapted from Wayne Gowen's album, Chronicles of Carmela. Special thanks to him for providing that to us. Something to Chew On is produced by the Office of Research Development at Kansas State University.